Hello everyone. In this class, we'll study about smooth muscles. Smooth muscle is distinguished anatomically from skeletal and cardiac muscle because it lacks visible cross striations. Actin and myosin too are present and they slide on each other to produce contraction. However, they are not arranged in regular arrays as in skeletal and cardiac muscle, and so the striations are absent. There are different types of smooth muscles. There is a considerable variation in the structure and function of smooth muscle in different parts of the body. In general, smooth muscle can be divided into visceral or unitary smooth muscle and multi-unit smooth muscle. Visceral smooth muscle. Electrical and mechanical activity of visceral smooth muscle. Visceral smooth muscle is characterized by instability of its membrane potential and by the fact that it shows continuous irregular contraction that are independent of its nerve supply. This maintained state of partial contraction is called as tonus or tone. The membrane potential has no true resting value, being relatively low when the tissue is active and higher when it is inhibited. But in periods of relative quiescence, it averages about minus 50 millivolts. The excitation contraction coupling in visceral smooth muscle is a very slow process compared to with that in skeletal muscle and cardiac muscle, in which the time from the initial depolarization to initiation of contraction is less than 10 milliseconds. Molecular basis of smooth muscle contraction. Calcium is involved in the initiation of contraction of smooth muscle. However, visceral smooth muscle generally has a poorly developed sarcoplasmic reticulum, and the increase in the intracellular calcium concentration that initiates contraction is primarily due to the calcium influx from the ECF via voltage gated and ligand gated calcium channels. In addition, myosin in the smooth muscle must be phosphorylated for activation of the myosin ATPase. In smooth muscle, calcium binds to colmodulin and the resulting complex activates colmodulin-dependent myosin light chain kinase. This enzyme catalyzes the phosphorylation of the myosin light chain on serine at position 19. The phosphorylation allows the myosin ATPase to be activated and actin slides on myosin, producing contraction. This is the contrast to skeletal and cardiac muscle where contraction is triggered by binding of calcium to troponin C. Myosin is dephosphorylated by myosin light chain phosphatase in the cell. This enzyme is inhibited when it is dephosphorylated and activated by dephosphorylation. It is dephosphorylated by Rho associated kinase that is activated by ligands which produce inhibition of smooth muscle activity. However, dephosphorylation of myosin light chain kinase does not necessarily lead to relaxation of the smooth muscle. Various other mechanisms such as latch bridge mechanism by which myosin cross bridges remain attached to the actin for some time after cytoplasmic concentration falls. This produces sustained contraction with little expenditure of energy, which is especially important in vascular smooth muscle. Relaxation of the muscle presumably occurs when there is a final dissociation of calcium colmodulin complex or when the other mechanism comes into play. The events leading to contraction and relaxation of smooth muscle are summarized below. Initially, binding of acetylcholine occurs to the muscarinic receptor, which causes increase in influx of calcium into the cell. Calcium binds to colmodulin. There will be activation of colmodulin dependent myosin light chain kinase, which leads to phosphorylation of myosin. Increased myosin ATPase activity and binding of the myosin to actin occurs that leads to contraction. Then dephosphorylation of myosin light chain by myosin light chain phosphatase occurs, which causes relaxation or sustained contraction due to the latch bridge and other mechanisms. These are some of the difference between single unit or multi-unit smooth muscles. Single unit are also called as visceral smooth muscles. It is called single unit smooth muscles because it functions in a sensual fashion. It occurs in large sheets and has low resistance. Uh, and single unit smooth muscles have low resistance bridges between the individual muscle cells. Whereas multi unit, they are called as multi unit because it is made up of individual units without interconnecting bridges, that is non sensual in character. So its contractions are discrete, fine, and they are localized. Smooth muscles location, they are usually in the walls of the hollow viscera, that is GIT, ureters, bronchi, uterus, and urinary bladder. Whereas multi-unit smooth muscles, they are present in the ciliary muscles of the eye, that is iris, pilometer, muscles of the skin, and muscles of blood vessels. Whereas this visceral muscle, in certain areas muscles are characterized by appearance of spontaneous activity, and multi unit smooth muscles fibers has its own nerve supply. And in single unit, rhythmic contraction and relaxation of these muscles is independent of their innervation, and innervation has a role only in modulating its activity, whereas multi-unit muscles sensitive to chemical mediators at the nerve endings, that is acetylcholine or norepinephrine, in response to which muscle contracts. Single stimulus to the nerve causes a repeated firing of action potential, which produces irregular tetanic contractions rather than a single muscle twitch as seen in case of skeletal muscle. In in single unit muscles, active tension is produced, whereas there is no response uh, is produced upon activation of the upon stimulation of the smooth muscle. These are the some of the differences between single and multi-unit smooth muscles and about the smooth muscles. Thank you.